So anyway, well, so we do have, so our speaker tonight is Professor Linden, and he is a professor of neuroscience at Johns Hopkins University. And he's got a wide-ranging intellect and turns the dullest topic into something wonderfully interesting. He is going to talk about pleasure circuits in the brain. He has a book. We'll talk your book for you. It's really amazing. Called The Compass of Pleasure. And this is Lloyd's copy. He wants some autographs. But no further ado, Dr. Linden.
The talks on pleasure, so I brought some alcohol. How's that?
Great.
All right, good. Well, thank you for having me here, and I'd like to tell you a little bit about pleasure circuits in the brain. And I'm going to start off by reading a brief bit from the page, and then we'll go extend brains in. Bangkok, 1989. The afternoon rains have ended, leaving the early evening air briefly free of smog and allowing that distinctive Thai perfume, French and pounding with faint note of sewage, to waft over the shiny streets. I hail a tuk-tuk, a three-wheel motorcycle taxi, and hop aboard. My young driver has an entrepreneurial smile as he turns around and begins the usual interrogation of male travelers. So, you want a girl? No. I see. Long pause, eyebrows slightly raised. You want boy? No. Longer pause, sound of engine, sputtering at idle. You want lady boy? No, I answer a bit more emphatically. I got cheap cigarettes. Johnny Walker? No thanks. Undaunted, he moves on to the next category of his wares, now with lowered voice. You want ganja? No. Coke? No. Yapa? Yapa is Thai for amphetamine tablets. No. Heroin. You want heroin? No. Voice is raised back to normal. I can take you to a cockfight. You can gamble. All right, I'll pass. Just a little bit irritated now. So, firm, what do you want? You knew I respond. Those little mouse shit peppers. I want some good spicy dinner. My driver, not surprisingly, is disappointed. And as we tear through the streets to a restaurant, blasting through bubbles, I'm left wondering, aside from various shades of illegality, what do all those offers have in common? What exactly is it that makes a vice? Now, there are a lot of ways to approach this question. You could approach it through anthropology or social history, because what makes a vice is certainly something that is culturally constrained. But I'm a biologist, and so what I'm seeking is a biological explanation. And a biological explanation is going to be cruder. It's not going to be as detailed. It's not going to have as much cultural analysis as an explanation offered by an anthropologist, but it has certain degrees of utility that are greater. It's more universal, for example. So I think the central thing that I want to convey is that we have a circuit for pleasure, a circuit for reward in our brains. And it's called the medial forebrain pleasure circuit. And medial is just a word meaning it's in the middle of the brain, and forebrain means it's towards the front of the brain. And it consists of several interconnected regions. The names aren't that important, but I'll tell you anyway. One main part is called the ventral tegmental area, and it sends information to another area called the nucleus accumbens. Another area is called the dorsal striatum. And the feeling of pleasure that is produced when these areas are activated is critically dependent upon the release of a neurotransmitter called dopamine, that I'm sure many of you have heard of. Now, how do we know that this area of the brain is involved in pleasure? Well, some of the first clues came in Montreal, Canada, for in the 1950s, when experimenters were implanting electrodes in the brains of rats, and they were trying to hit a region of the brain that they thought had to do with attention. And they missed,
and instead they hit a part of this glucocorticoid pleasure circuit. And then they had this electrode was hooked up to a circuit that could deliver a mild electrical shock and activate that part of the brain, mimic its own natural activation, so to speak. And they had the rat in a big box with 4 corners labeled A, B, and C, and D. And if the rat was an A, and they delivered stimulation to this brain region while it was an A, well, then the rat would really get interested in A and explore A a lot. And if they did the same thing for B, well, then it became very interested in B, interested in what's going on here. And then they did something clever. They, instead of having the experiment return the switch, they put a tiny palm-operated switch over each stage. And they allowed the rat to self-stimulate its brain. And what they discovered was that these rats would beat on the switch like a 4-year-old playing Donkey Kong. They would mash that switch constantly. As a matter of fact, they would do it at the rate of 7,000 lever presses per hour. They would do it to the point that they would starve themselves to death. They would do it to the point that mother rats that had just given birth to a litter of pups would ignore their pups, let them die so that they continue starving. They would do it to the point where they would cross electrified grids in the floor of the cage and get painful shocks in order to get this pleasure. So this was an extraordinarily strong motivator for these rats. So you're probably thinking, well, what about people? And so, well, there are ethical concerns in doing experimental things, aren't there? Well, there are a few examples. These kinds of experiments aren't done very much anymore, which I'm happy to report. There were some examples where, like the rats, like the initial rat experiment, the electrodes came to rest in the wrong place. In other words, somebody was implanting an electrode that might be useful for the relief of chronic pain, and there's a legitimate neurological use for that, and it missed, and it wound up at the pleasure center. In that case, the human subjects behaved just like the rats. All they wanted to do was to press that button all day long. Sometimes if the button was set so that you could only press it every 10 seconds, they'd start taking it apart and tinkering with it and trying to get around it. There also were some very unethical experiments that were done at Tulane University in this. And so, for example, there were a pair of researchers who believed that homosexuality was a sickness and that you could turn gay people straight by having them watch hetero porn while you were stimulating the pleasure centers of the brain. I mean, this is a reprehensible experiment. And astonishingly, it was approved by the Human Subjects Review Committee at Tulane. It went on for years. Amazingly, one of the lead investigators was named Dr. Moan. And sometimes your name is your destiny. So Dr. Moan and Dr. Heath hooked gay men up to, they had surgery to implant these electrodes. They had them watch hetero porn and stimulated their brains and asked them, well, did you become, sexually excited? And they said, well, yeah, kind of, I don't know, it felt kind of good because, the pleasure centers were being activated. And then, so they wanted to know though, but like, did this really work? Did we actually turn this guy straight? So they thought very carefully about the correct experiment to do, and then they went out and they paired a hooker. And they brought the hooker into the lab, and they had this poor fellow with all the EEG wires attached to his head. And yes, the hooker was able to seduce him, and they had sexual intercourse, and he ejaculated, and they wrote a paper about it. And in my mind, this goes down as one of the most heinous experiments ever published in the neurobiological literature, and it's an embarrassment to all the brain researchers in this room. All right. So what's the pleasure circuit for? Well, one clue is that it's evolutionarily ancient. So these structures aren't just there in humans, they're not just in primates, they're not even just in mammals. You can find them in birds and reptiles and amphibians, for example. And basically, this pleasure circuit is there so that we eat food and drink water and have sex, so that we survive and pass our genes on to the next generation. And so it's really straightforward. You want those things to be pleasurable if you want to succeed evolutionarily. So how do we know this? Well, so for example, if we were to take someone here in the audience and put you in a brain scanning machine, and let's say you haven't eaten for a while, you'd skip the block of chips in the back, and you were real hungry, and we gave you a bite of one of your very favorite foods. And if we were using a brain scanner that could detect this, called the positron emission of the scanner, that could detect dopamine release within the nucleus accumbens, we could see that amount when you took that first bite. But what's interesting is that not only would we see that activation of the pleasure center in this way, but your subjective experience of pleasure would be tracked. What do I mean? Well, so you know when you're really hungry, the first bite tastes great, and the next bite maybe not so much, and maybe the last bite you eat before you stop is the least pleasurable. And if we look at the amplitude of dopamine release in the situation, it would follow your subjective experience, with the first bite giving the largest dopamine release, and then gradually smaller and smaller as things progressed. Likewise, if we give you food you really like, big dopamine release. A food that you're indifferent to, small dopamine